Hello, my name is Prajakta Yadav. I am a student of Britsung Training Center. Today, I am going to discuss a technical topic about signal detection in pharmacovigilance. So let's start with the topic. Introduction to Pharmacovigilance. Pharmacovigilance refers to the science and activities relating to the detection, assessment, understanding, and prevention of adverse drug reactions or any other drug related problems. Its goal is to improve patient safety and ensure that the benefits of a drug outweigh the risk. A critical component of pharmacovigilance is signal detection, which is the process of identifying potential safety concerns about medicines based on collected data. What is signal detection? Signal detection is the method through which new or previously unrecognized safety concerns, referred to as signals, are identified in a drug safety surveillance. These signals may suggest that a drug is associated with previously unknown adverse effect or a higher frequency of an already known adverse effect. Identifying these signals is essential to take appropriate regulatory actions, which can range from issuing warnings to withdrawing a product from the market. In the context of pharmacovigilance, a signal is identified as information arising from one or multiple sources that suggests a new potentially causal association or a new aspect of a known association between a drug and adverse event. Sources of data for signal detection. Signal detection relies on various data sources which include spontaneous reporting system. These are the primary sources of data for pharmacovigilance. Healthcare professionals, patients, or manufacturers report adverse events voluntarily, and this information is collated in national or international databases. Example, the FDA Adverse Event Reporting System, that is FAIRS, whose VG-base clinical trials during the pre-marketing base phase. Clinical trials helped detect ADRs. However, their ability to capture rare or long-term side effects is limited due to the controlled and short-term nature of trials. Literature and published reports. Published articles, case reports, and meta analysis in medical journals can also serve as sources for signal detection. Electronic health records. Data from healthcare systems or patient registries, especially when they include large populations, can be useful for post marketing surveillance. Health insurance claims and billing data. Large databases from insurance claims can help identify associations between drug usage and adverse events. Methods of signal detection. Signal detection in pharmacovigilance generally involves two main types of approaches. Quantitative methods. These methods rely on statistical techniques to detect signals by analyzing large data sets. For example, include proportional reporting ratio. The PRR compares the frequency of a specific adverse event with the frequency of all other adverse events in a database. A high PRR suggests that the adverse event is more commonly associated with a specific drug. Bayesian data mining. This method uses Bayesian statistical technique to identify drug event associations by comparing observed and expected numbers of reports for a particular drug event pair. Reporting odds ratio. Similar to the PRR, this method evaluates the odds of a drug being associated with a particular adverse event relative to other drugs. Qualitative methods. These methods involve expert review of available data and qualitative analysis such as narrative reviews of spontaneous reports or signal detection algorithms that use case reports and clinical expertise to suggest potential signals. Steps in clinical detection. The clinical detection process generally follows these steps: data collection, data analysis, signal prioritization, signal validation, risk assessment, regulatory action. Challenges in signal detection. While signal detection is a crucial part of pharmacovigilance, several challenges exist in this process are following: underreporting of adverse events, data quality and consistency, causality. These are the challenges in signal detection. Today we discussed about the signal detection in pharmacovigilance. Thank you for listening.